So today we're going to talk about microbes, and that's pretty cool. By microbes we mean microorganisms. All right. So what's the point? Today you're going to look at different types of microorganisms, and at the end you should be able to recall and tabulate key properties of these microbes. So your microbe will be the independent. This means it'll be in the left-hand column, and all the properties to the right of that. All right. So these are the ones we're looking at. We're going to look at bacteria, viruses, protozoa, and I'm going to mention fungi. Now, no, not fungi, sorry. We're going to look at fungi in a bit more detail. I'm going to mention prions. Prions are weird. Prions are a disease that's caused by prions is mad cow disease, or the human version of that is called Creutzfeldt-Jakob disease, or CJD. CJD is caused by an irregular protein that cycles around the body. Now, you normally have the protein, but a mutation to it can cause it to be a slightly different shape, stops it from working, causes all sorts of problems, and then can be passed on to other individuals. All right. So you're going to take notes Cornell style. All right. Then you're going to process these notes at the end with the headings: microbe, size, so into a table: microbe, size, replication, DNA, example of disease, treatment, and a fun fact that you're going to find on the internet yourself. All right, so bacteria. They are ubiquitous. This means they are everywhere. And for every 5 grams of soil, you've got about 1 billion bacteria in there. So they cover your body, and they're a really important part of your digestive tract. They make up about 50% of the Earth's biomass. So this thing that we can't see, if you could get some big cosmic scale, you put everything that's alive on one side and just the bacteria on the other, and the bacteria would probably be a little bit heavier. All right, they're unicellular, so they have one cell for each organism, but they can survive by clumping together. They're about 0.5 to 4 micrometers in size. Now, a micrometer is a millionth of a meter, or 10 to the minus 6 of a meter. They reproduce asexually by binary fission. Now, there are other ways they can do this, but that's the key way. All right, so this means that one cell is identical to its parent cells, or unless there's a mutation, but we'll get further into that when we cover antibiotics. They have no nuclear bound organelles, which means the DNA is not in a nucleus. They're free. It's free DNA floating as plasmids, which are rings, and they're more complex than that, but they float freely in the cytoplasm. Very helpful, as we said, in digestion, also controlling disease. Like you've got bacteria all over your body, and that's helpful to you. It secretes acid, which means that other bacteria can't survive in your skin. So that's pretty cool. Diseases do include food poisoning, syphilis, tuberculosis, which is a major problem at the moment throughout the world, and the treatment is antibiotics. Now, we will cover in another lecture, just down the road, how we need to be careful with antibiotics. That's actually a scary thing for the future. All right, so this is a model that shows us how they replicate. So the DNA copies over here. That's cool. Then we have what we call invagination, where the DNA is copied and it separates, and the cell wall just turns in on itself. It makes a cavity, and then eventually, when that's complete, it breaks apart, and we get 2 separate cells that have identical DNA, 99.9999 whatever percent of the time. All right, fungi. Now, there are very few pathogenic fungi. They tend to be surface diseases, and they're what we call opportunistic pathogens. In other words, they might live there normally and just be cool, but it's only when your immune system or a bit of free space opens up, like the bacteria dies off or your immune system weakens, that they really tend to go nuts. All right. Generally, they're decomposers in our ecosystems. They're ultra important. If it wasn't for these guys, we'd just have dead things lying around the place. They're unicellular, but they often, most commonly, exist as colonies, so they exist in clumps together. A mushroom is millions of different organisms bound together in a colony. They're between 5 and 25 micrometers, so they're larger than bacteria. Some can be seen with the naked eye, but yes. All right, so They reproduce asexually by budding and binary fission. And this over here, this is budding. So the DNA comes off to the edge here, the nucleus, and it copies. So and a little separate bud appears, and the DNA replicates, goes into there, and then it's all separated. It's cool. And then you've got this smaller one off to the side, and that grows. All right. So, and we see just here, this is a footage of it happening in real life, in real time. All right. So that's not real time. That's sped up heaps. I don't know why I said that. They have nuclear DNA. It's in the nucleus, as we see it here. That's why it's drawn as this little circle. Very helpful. They decay organic matter. And diseases, things like tinea, thrush, candida, albicans. So generally yeast infections. Now the treatments are antifungals, so topical creams, and we can saute with butter, like a good mushroom. Be tasty, tasty. All right, protozoa. So these are most common in tropical and subtropical regions. We tend to consider them an animal, to be honest. Well, not really, but they're very similar to animals. They require food. They don't have cell walls. Except they're unicellular. And we see it over here. The protozoas are chowing down and having a feed, and that's pretty cool. So they engulf it. All right. They require a host, and they often have complex multi-host life cycles. And we'll see one of those in a minute. It's quite intense. They're between 1 and 50 micrometers for human pathogens, but the largest is about 150 micrometers. So that's pretty insane. All right. They reproduce both sexually and asexually, and this means you actually get a lot more variation. They require a wet environment. They don't live in the dry. They need it to be a liquid medium. So this is where they might cause real problems in humans. Is that they live in your blood, and yeah. All right. So nuclear DNA in the nucleus. Well, so there's your DNA. And diseases include malaria, which has killed half of the people alive today. Not half of the people who have ever lived have died from malaria. Well, so it's estimated. And you've got Giardia and Cryptosporidium. Now, for those of you who live in Sydney, you might recall about 7 or 8 years back, there was a pretty serious water contamination, and that was Cryptosporidium and Giardia. All right. Treatment. We use antiprotozoal agents, so that's a chemical that goes in and kills them off. But recently, they're not very effective, so recently we've been using viruses to to infect the protozoa and kill it off. All right, so viruses. Now, viruses, they're not considered living organisms. They have no cell, they can't reproduce on their own. They require a host. They're very, very small. They're between 10 to 750 nanometers. Now, that's 1 nanometer is 1,000th of a micrometer, or 1,000,000,000th of a meter, 10 to the minus 9 meters. So they reproduce by hijacking the host cell's machinery, and it kind of looks like this if this video works. All right, so we'll give that a second. All right, so the DNA or RNA, it can have either, is in a protein capsid, and you see that over here up on the top. Okay, so here we go. The virus, and this is a real virus. Like it looks like this. It comes along, and that's DNA there, genome, and it injects itself into the cell. This is a bacteriophage, so this actually affects bacteria. Injects the DNA in, and it then uses the equipment in the cell, like the chemical equipment in the cell, to replicate it. And so then here we go, and now it's going to put itself all together, and then breaks apart the cell and releases to spread into the neighboring cells. So that's viruses. So diseases include influenza, pertussis, which is whooping cough, which is something we're going to talk about a lot in a future lecture on vaccinations, and HIV. So treatment. There are some antivirals, but viruses are pretty hard to treat without causing significant damage. So usually we leave them to run its course. The way we kill viruses tends to be by killing the host cell, which means your cells, and that's often the cure is worse than the disease. All right, that's it. That was a long one. I hope you're able to keep up. See you in class.